we're going to take a little bit of a turn from, I realize all these talks are so beautiful, and I realize why I love math, but I also realize I don't do math. At the same time, I was a math undergrad, so it was really nice all these talks. I'm going to start today. I call this talk Statistical Opportunities, which I think you can think about this as maybe statistical opportunities, mathematical opportunities, how you want to think about it. Really is a broader question of thinking about how to define and target cell state in cancer, but you can also maybe reposition this as thinking about statistical opportunities in the age of generative AI, because I'm going to speak a lot about that today. But before I begin, you're probably wondering why is someone from Microsoft here talking about bio? So I figure I'd tell you a little bit about Microsoft research. So MSR is a global organization with actually many different things that don't always have to do with just computer science. There are people who study anything from social technical systems, so social scientists really trying to understand the intersection between race, technology, and communities. And they also have a large health sciences and life organization arm called Health Futures, and I'm part of this group called the Biomedical Machine Learning Group, which is a subset of that. So the Biomedical Machine Learning Group kind of spans us in Boston, but also there are people in real Cambridge, UK, and then also people hanging out in Redmond. The team is actually quite diverse. There are people, not just myself, who I do a lot of genetics and genomics-based work, which you'll see today, but also people thinking about questions like Kevin Yang on thinking about protein language models, people like Kirsten Severson thinking about imaging technologies, and then you have people like Alex Liu and Ava thinking about everything kind of in between, which is really nice. And so we work across different multiple scales. And so the real big question at MSR is really thinking about how to use generative AI to really understand questions both at like the molecular scale, cellular, tissues, and across the patient samples, but also to leverage data across these scales, combine them in a way that we can build better generative AI systems. And so kind of this two-way kind of relationship. So a little bit about me. So my background is in statistics. I typically think questions about how non-additive variation plays a role in complex traits and diseases. The way you can think about this is a trait can be broken down into basically three different components. There's an additive component, which is the idea of how do you take gene A's effect plus gene B, and how does that relate to a given trait? There's this nonlinear component, which is the idea of what is gene A's effect times gene B, or gene A's effect times something going on in the environment, and how does that play a role into a given trait? Trait here could be anything like height, body mass index, something more complex like schizophrenia. And then there's a big component, which is what role does the environment on its own play, right? And so I think about the breakdown of these different traits across different populations of people, right? And when this genetic architecture is how we define it, when a genetic architecture is the same across everybody, let's say in this room, precision medicine is really easy, right? Because then I can just make one drug and that'll work for everyone. In the real context, it's a little bit more complicated, which is this question of what happens when this genetic architecture might change for different populations of people. And this population breakdown could be anyone with different ancestral backgrounds, but also anyone, to different age groups, right? In that case, you want to understand what's driving these differences across different populations of people, and is that something that we can overcome by creating better drugs? Now, today's talk is going to be very similar, but instead of thinking about global traits for humans, we're going to think about this on a little bit of a different scale across tissues, right? It's the same kind of question, how to additive effects, but also non-additive effects. And non-additive here is going to be, gene by gene interactions, but also could be something like a gene by microenvironmental interactions of this given cell. How does it then contribute to things like therapeutic resistance in cancer? Okay. So my background, again, is statistics. When I was co-advised during my PhD, I was co-advised with someone in pharmacology cancer biology. And so there was someone yesterday that made a joke about how they don't pipette. That's also my claim to fame. I also don't pipette. I do understand how to do those experiments, though, which is really, really nice. And so we're gonna hang out a lot in this cancer space for the next couple of minutes. Okay, cool. So now to get into the crux of the talk. So, for those who do not study cancer, DNA is kind of the cornerstone for how we think about cancer therapeutics and precision medicine in this space. And what I mean by that is, for a lot of cancers, we understand the driver mutations as things that happen kind of on a DNA scale, and we understand how those might differ across different patient backgrounds and certain patient populations, right? That's also the key, DNA, being of how we think about how to do drug discovery a lot of times, right? So you might have some type of model, some ex vivo model, whether that be a cell line, an organoid, some patient-derived xenograft. And what we typically want to do is, if I understand the distribution of DNA mutations in a given patient, what I try to do is I try to match that distribution of patient DNA mutations in a dish that I would see in a patient. And if those two things kind of align, then I basically say that this is an okay representation of what is going to go on in terms of, if a drug is effective or not, what I should see also be mirrored in the body, okay? And so we can think about, driving these type of models to think about, different patient stratifications, right? And so I can use DNA to basically match patients on the therapy. So those who don't know, like I might have a certain set of mutational profiles that I know might have worked for a given drug study. I look for the patients that kind of have the alignment of the same mutations in the population. Those individuals, I consider those maybe actionable. I'll put those on a given targeted therapy, right? And I'll put the rest of the people don't get put on these different programs, okay? Now, the key to this current paradigm is that this only works if DNA is actually like the driving force. So in other words, that drug response in cancer therapeutics is like mutationally encoded, okay? Now, there's some cases that we have wins for this, right? So there's some successes. Some successes of here are like HER2 amplified breast cancers. You have a lot of leukemias and lymphomas where this has been successful, where I understand what's actionable, what's not actionable, and so if you do have a mutation, I'll put you on this drug, okay? And there are a lot of cases where this is not actually the case. And so the reality of this situation is that this actually only captures about 40% of patients, right? And what's even worse is that out of that fraction, only a fraction actually see some sort of initial success when they're put on these drugs. And what's even harder is that only a fraction of that fraction don't see experience things like therapeutic resistance, right? So only a small actual proportion of the individuals have this actually work. And the reason for this is because, they're limited responses due to both heterogeneity and or development of resistance that can occur both because of genetics, but also because of non-genetic driven mechanisms. And so really what we're trying to ask here is like how many dependencies are actually explained by just single features in the body. And so if we think about this as a total paradigm, cancer cells have very diverse inputs, okay? So where we typically stay now is, we have DNA, which I just said was kind of is a static kind of a representation of something that's static, mutated, and that's kind of our blueprint. You have RNA, which is actually really interesting because RNA is a continuous expression, right? But it also kind of tags, or in other ways, you can think about this as like correlation. It's sort of correlated in some ways where it kind of captures both what's happening on the DNA scale, but also gets a little bit of inside of its measurement, what's happening epigenetically, as well as interactions in the environment. And so you have more of like this continuous measurement, right? And RNA as this kind of like RNA state or something I'll define in a second, all kind of contribute to this like diverse cell attributes that you might see, okay? And so, cell state, as you're going to hear me say a lot, is kind of like this idea of group of genes co-expressed by cell mRNA, capturing both cell function as well as environmental context, all right? Now, when we think about drugs, right now, drug development, all our current pipelines remain almost exclusively here, right? And what we're going to ask is, can I come up with like some sort of cell-based or state-based lethality program where I can start to target drugs, try to find drug targets that kind of holistically look at the cell overall, right? And more in this like dynamic interpretation. And so really we're gonna ask, like, do we have great tools for this, and how do we kind of measure or quantify more complex cell state attributes? Okay, so for this talk, some people have mentioned this yesterday, we're gonna do a lot of stuff with what we call single cell readout, okay? So bulk expression is what you might have heard in the past, is that this idea that if I have a tumor, I'll get a biopsy of that tumor. I can sequence it, and I'll get this kind of average expression profile where these different cells here are different, or these different blocks here, maybe not to confuse it, but these different blocks here are different genes. And this average expression is going to represent all the expression across all genes and all cells in this given sample, right? This kind of averaged out thing. Now what we can do is we can actually sequence individual cells that make up this given biopsy, right? So we're going to get now these like large matrices, cell going from 1 to n. N in this case for a lot of data sets now, or can be up to hundreds of thousands of cells for a given experiment, or all the way to like 1 million cells, and we'll understand more of like the heterogeneity that happens across different cell types. All right, and what we're going to do is we're going to do like integrative approaches to measure and understand biology, kind of at the single cell resolution. Now, in the past, this was like kind of hard data to get, but we're kind of in this really nice regime now, where these data sets are kind of in abundance. And so you have these nice cell atlases that are being publicly produce these things like the Chan-Zuckerberg initiative has done a lot of this with this data source called CellXGene. And there's also resources coming out where people have been doing a lot of high-scale, high-throughput perturbation experiments, like the Tahoe 100 million cell project, where all these kind of data sets are available. The reason I really like this, to show this to people, is the stuff that we work on is both equal parts computation and biology. And so let's think about a really small test where I have, let's say I'm only able to capture 500 cells. Well, I might run that 500 cell experiment across a panel of 225 genes, which we know are commonly mutated in this given tumor case. I can get RNA expression for 20,000 genes, and I might think about how to capture both this idea of cell by gene interactions, but also cell to cell interactions. And just for a small study, I might be, or per biopsy, I might be able to get something like 44 million data points. And so our data sets can be quite massive. And so I think this is a really cool problem where you might think about also the experimental design part of it with respect to biology, but there's a lot of computational questions that also come to play. And then, so this is what Project Ex Vivo kind of does. It's kind of this idea of bridging biology between machine learning and AI, as well as with experimentation. And so this is actually a co-led project that is ran by both Microsoft, the Broad Institute, and Dana-Farber. It's co-led by myself and Ava Meany on the Broad side, and then Peter Winter and Sri Raghavan on the Microsoft side, and Peter and Sri on the Broad side. And we have a great team of people both in computation, but as well as experimentation. What kind of makes this thing kind of go is this idea that we're all kind of co-located by each other. So we all are here in Kendall Square. Microsoft Research is down the street from the Broad Institute. And so how this works logistically is we actually get patient-derived samples from Dana-Farber that then are sequenced at the Broad Institute, and then we upload and do everything via like Azure at Microsoft Research. For those who haven't been to Boston, this looks close, but this river actually can make things quite far because the train doesn't go that way. So there are going to be two parts to this talk that I'll kind of focus on. The first is the idea of fidelity of cancer states. And the second is I'll talk about some work that we've been doing a lot on, which is the idea of how to build foundational AI models and state-to-function maps in this space. And so, how does the environment affect cell state and therapeutic response? And you might actually just ask, like, why do I even care about this cell state problem? And here's actually a really cool example of this. So this is from a Cell paper that we recently did on pancreatic cancer. Pancreatic cancer has a few driver mutations that we know to be prevalent across many individuals. And remember what I said before. What I might do in a lot of cases when I think about building models ex vivo to model what happens in given patients is I want the distribution of mutations that I observe in my ex vivo model to match what I would see in terms of distributions in terms of mutations I would see in a patient. Now here's the kicker. All of us can have the same driver mutations that are present in our given biopsies, but we might have different cell states that are represented. And so what that means is something like this. So this is what you see in terms of gene expression, and this is actually why a lot of therapeutics that look like they work in the lab don't transfer over to working in patients, is because I can have the same distribution of mutations, but here's in terms of different cell states. So here's two classical ones where they have classical cell state and basal, which are quite prevalent in pancreatic cancer. And here you see like a difference of for this given set of individuals, these are more classical patients, and then these are more basal individuals. And you can see what happens in just my cell line. I just represent those that are basal, or that have a basal type phenotype. And so if I then decide that I'm going to go do transfer my treatment that I thought worked here to the patient population, I'm not going to recapitulate the same sensitivity. And why this actually happens is because you actually lose microenvironmental factors that make humans human when you grow things in a dish. And so one really cool study that we kind of showed is that, well, if I can identify the things that I lose in a dish, so in this case for pancreatic cancer, this will be TGF-beta signaling. I lose that, so I lose my ability to identify the basal signature. If I can add those cells, that cell state signature back into my dish, I can recover those cells that I was missing for more models that have higher fidelity that then match what I would see in vivo. And the cool part about that is we showed that, typically what you have is this like the non-correlation effectively between what you see in a response from a lab model to what you might see in a matched in vivo patient model. And when you add these state signatures back in, you kind of recover this response. And so we learned a lot about this, and this idea that you can almost leverage cell plasticity against it if you can identify these things computationally. And here's a really cool example of how now we're doing this, and this will make a full circle later in this talk. But one thing that we're doing, we actually have wet lab space at the Broad where we can actually do this, is you can actually engineer state heterogeneity in cancer cells. So here's a setting where I have a bunch of what they call open read frames. I know a set of transcription factors that I know are kind of key drivers in certain mutational states. So what I can do actually is, in a given experiment, I can take these ORFs, I can apply them to a transcription factor, and I can push cells to express the signatures or be of the cell state that I actually want them to be. So I can almost make data sets that I actually want in the lab if I know what I'm missing in the lab. So here's an example of this. Here I'm missing the classical state signature. We take these ORFs, we overexpress these different transcription factors, and I can drive these cells to have more of a certain state signature. And we can do this across many different transcription factors. Here's a panel with 50 of them, and I can identify which of these kind of drive me to different states that I know that I might be missing. You'll see later in this talk for certain models or whatever the case might be. And so we have this really nice connection where computational I can make the data sets that I want to learn from. So cool. So we've been doing a lot of computational stuff in this space. And so, you know, anything from more simple, and then we'll get more complicated on. You know, things that are simple that we've been doing is thinking about things that are typically fraught in the way that we do things bioinformatically in single cell. So here's a really cool example that people have been working on recently. Clustering is actually a really big step for people to understand like what cell types are in your data set. Cell annotation is like a key task that you want to do whenever you get new cells that come off the sequencer, what cells are present in my data set. Now the way that people do this is, typically what you do is you take a data set, you cluster that data set in some way, and then you ask yourself, well, which are the differential expressed genes between these clusters, right, that are basically signatures of these, that makes things different between these two cell types. If you're a statistician, you're probably like, this is insanely wild. Why would you do this? And the reason for this is because this is a classic double dipping problem, right? Like, if I cluster my data set, then I ask myself, what is different between the clusters? The answer is everything. So like, why? Like, so there's a huge type 1 error and false discovery rate process that happens when you try to identify differentially expressed genes. The answer is everything, so everything, all your test statistics have to be really inflated. So we've been doing basic ideas of like how to maybe overcome this. One clear example of how we do this that's actually really simple is this idea of calibrated clustering. And this calibrated clustering that was led by a student in my lab, Alan, works in this very simple way. So the calibrated clustering idea is really based on this idea kind of similar to what knockoffs are, if anyone's familiar with this in statistics. So what recall does is for every single gene in your data set, you create basically this artificial gene. So I have like a generative model for which I can simulate single cell data. I'll take a real gene, I'll create a fake gene for every real gene in my data set. The second step here is what I'll do is I'll concatenate both the real gene expression data with the fake genes, and then I'll perform clustering on this concatenated data set, right? Then once I have all of my clusters, what I'll do is I'll do my differential expression analysis. So I'll get p-values, but for every cluster comparison for all genes, both the real and the fake ones, right? And so I have a p-value for every jth real gene and a p-value for every corresponding synthetic gene. And then I'll create these contrast statistics comparing the different p-values. And the idea is simple, right? If my fake gene is more statistically significant than my real gene, I've probably done something wrong where I've over-clustered in my data set, okay? And so we'll take this test statistic, or this contrast, and I'll basically ask the setting for which this contrast statistic is over some allowable threshold. For here, we'll set this threshold to be 0, for which I'm saying I never want any of my real genes to be less statistically significant than my real one, right? Or my fake ones, right? And so we could do this, and what recall will do is say, well, if that situation ever exists, then what we'll do is we'll basically say you've over-clustered your data set, you should recluster, but with fewer numbers in your algorithm, right? And you'll kind of continue this process until you have this calibrated setting where this situation never actually happens. And so the real reason for this happens because a lot of clustering algorithms in single cell, because of the small variation that happens in data sets, is you actually get this situation a lot. So let's say this is a simulated data set. Let's say I take a simulation where I have my true labels is all my cells are all of one type. If I run a classical algorithm from software that people typically use, I might get a situation where I have mini clusters. And what recall will do, we'll take these mini clusters and say, well, you've actually over-clustered, and so you should go back to having just one. In a more realistic scenario, what you'll get is something like this, where I'll have true labels, and what a classic algorithm will do, will find small variation for which it'll start to separate certain cell types. And what recall will do will kind of help you from making this overcorrection, and it'll correct that for you so that you go back to the right number of clusters. So we do this across many different types of, against many different algorithms to try to solve this problem. The cool thing about recall is it's really simple. Like you just have to do this synthetic generation, and so it's quite fast to do, and it's actually really quite simple. And it seems to do better more robustly across all the methods. That's not really the point of this thing that I really want to get out of this for everybody, but the point of it is like the downstream analysis of how this kind of helps things. And so if I really were to take a simple clustering algorithm, so this is a real gene ontology, real cell ontology project from the Human Cell Atlas, where we have different cell types here that have been humanly annotated. Here's something you might get from a classic algorithm. And so you might see this like stark variation here. And so I might think that these cells in cell type 1 and cell type 2 are quite different, but if I look at the marker genes that are driving them, they seem to have very shared things. And what recall kind of does is kind of fixes this for you so that you never kind of make this kind of mistake to think these might be two different cell types. And so downstream, your idea of like what marker genes are driving different cell states are a lot clearer. I have many thoughts about this, but for the sake of time, I'm happy to talk about this later. And so the cell type annotation story actually is really important because actually this is a key task that we ask a lot of AI models to do, right? Now, AI models actually are not that great right now, so I'm not going to be AI enthusiast. I'm going to be very realist in the rest of this talk. And so cell type annotation, this idea that like I have a bunch of cells, I train a model on a certain set of cells with their annotations, and then I then try to predict when I see new cells what those annotations should actually be. And this is key because again, all my downstream things are kind of predicated on this labeling. There's a key challenge of why this is really hard to do in single cell biology. Right, so I might take a training set. So here's a training set of 15.2 million cells from, again, the CellX Gene Atlas from the Chan Zuckerberg Initiative. We'll train a bunch of different models on these. I might train something that's like a very simple linear model. I might train an MLP, some neural network, and I might have something that's a little bit deeper, this thing called TabNet. What you might see is that when I train these models, I'll train these models on these given labels that came from this data set. If I test something that's in distribution, meaning that I take a set of donors that have very similar cell annotations to the one I observed in my training set, all of my models tend to do really well. Actually, even something as a linear model. I don't need something that's really complicated to do this. It's a very simple task. The issue is when I move out of distribution. And then you'll see this like kind of uniform drop in performance as I move out of distribution. And the reason why this happens in single cell a lot of times is because your train test splits are typically split up by like a temporal setting, right? So my training sets might come from an Atlas release that happened, let's say, in late 2024, and then my test sets might come from data that's released in late 2025. Well, the issue because of this is that I might, as a field, we get more sophisticated in our ability to label. And so what might happen is that I have really coarse cell type annotations here. And then when I try to go in and try to make predictions on new cell type annotations, because we've gotten much more specialized, this model was actually now well equipped to be able to do that task really well. And so you see this massive drop in performances. Well, that makes a really cool, interesting point of like, well, how do you actually improve AI models for biology? And to me, there are three ways to kind of think about this. One is architecture, so the model architecture itself, right? One is the training objective, what you teach your model to actually do. And the third thing is the data that you put into it. A lot of people spend a lot of time on the first, but there's a lot of momentum that you can get by spending a lot of your time thinking about those latter 2 points. And I'll show you an example here. So here we're going to rethink about how we think about our training objective. And if you think about it, a lot of these models are trained using what we call cross-entropy loss, right? Effectively, that just means that like if I make a prediction of a given cell type, right, then, all these cell types will kind of sum to 1 in their probabilities, but I don't give the model a lot of wiggle room here. You either predict whether or not you're looking at a T cell, or if I'm looking at something that's way more complicated, or way more granular, right? An alpha beta T cell, a gamma delta T cell, and if I get it right or wrong, right? If I guess this coarse grain or something with more fine grained, doesn't necessarily matter. But because of this loss function, right, I get penalized in a uniform manner. But we know that cells are related in some ontology, right? I know that certain cell types are if I think about a tree, right, certain cell types are children of certain labels, right? And so one way to kind of, give the model a little bit more wiggle room is to not have it be so, it has to be this or that. You allow it to maybe not have to over-index on things. And so one way to do this is we thought about this like hierarchical cross-entropy loss, which is effectively this idea that, if you assume that it's a T cell, but you also assume that it's a gamma T cell, a gamma delta T cell, or an alpha beta T cell, this is all kind of in the T cell family. So don't make the model pick one, kind of allow it to say that in this region, right, this all is generally correct, right? And there's a way that, like, I might get data sets that have more granular levels, and I might get some that are not, right? And when you get this more wiggle room in your modeling, you're actually able to recover a lot of that performance that was actually lost, right, when I move to this out of distribution setting. So that's one kind of way to think about when you build models. And again, what I also kind of like to point out here too is that you can also improve linear models just as much as you improve something that's more deep in like a transformer-like thing, right? So this is a way to kind of ultimately improve your modeling ability, right? Not even changing architecture, just thinking about its ability to learn like what might be right and what might be wrong, right? So that really leads me to like my next part of the talk. We did this across many different cell types and many different data sets. And so what you're seeing here are the different cell type annotations. I mean, some of these go from being as broad as like, T cell, to something that's like super specialist in terms of like the way they were annotated. And you can see when you get this wiggle room, green here means improvement for a given cell type. Gray means that, we've kind of equated what was done with the regular cross entropy, and then red means there was a slight dip in performance. And here you can kind of see for this data set how much performance gains you can do by just looking at this particular alternative of a loss function. And we can do something that's really more cool. We can go back into like the cell ontology tree and then see for which cells do we do better than and you can see for a lot of them, it ends up being these leaves that are highly connected to other different cell types. And so one thing we kind of write in this paper below is to say, like, not only is this a computational thing, but hopefully it also motivates biologists that the more connected this tree is, right, the more powerful our models might actually become. So data, again, here is actually quite important. And so for the last like 30 minutes, I really want to spend time talking about like our latter half, and hopefully this also motivates this latter half of really thinking closely and deeply about what might power AI in this given space. And so really our group has really been spending a lot of time thinking about how to build foundational AI models and really in doing this, creating new data sets that allow us to understand what is a cell state and how does that then map to certain functions, right? And I've been asking a deeper question, which is like, can we even learn a foundational AI model of cell state, and like, what does AI over the space even mean? And so for a little primer on machine learning. So, foundation models, sorry if you hate that term, are supposed to enable diverse downstream tasks, okay? So what this means is that you take a data set, you train the data set on a given model, pick your favorite architecture, we'll talk about a few. What you can do is you can do one of two tasks primarily, right? And we'll break these down into different classes of tasks. One, we'll start with the bottom one, is zero shot. So when I say zero shot, what I mean is that I've taken a data set, I've trained a model with it, I freeze that model, and I use it for a bunch of different things, even if it wasn't trained for that given task, right? There's no updating of the weights are what they are, right? So foundation models, you can think about these things be like GPT, ChatGPT, whatever number you use on your phone, right? This idea of like, it was trained on a given set of data, the weights are frozen, and then you interact with it, and you can do everything zero shot. You can also think about a case where things are in the fine-tune regime. It's a little bit different. I've taken a data set, I've trained it on a given AI model. My AI model has its weights. I have a prediction task that I care about. What I do is I take a little bit of data that's in line with that prediction task. I update my weights a little bit with respect to that data, and then I do my prediction on that given task, right? So that's the idea of like fine-tuning. You like fine-tune your model a little bit, okay? Now, you can ask yourself, like, which of these two regimes really makes a model foundational? You can imagine a setting where both prediction and zero-shot discovery capabilities are actually really interesting, right? And I can come up with, we'll talk about a few in a second. Now, how do these models actually work? So typically, masked language modeling pre-training, like that objective is typically what's used in a lot of methods, particularly ones a lot interactive for language, right? So let's take a sequence, like a set of text. And this is a basic rendition of how these things work. I typically take this text. What I do is I break it up into what we call tokens, right? So I take each sequence, I have that sequence, parts of that sequence represented by a given token. Then what I do is I mask out parts of some of those tokens, okay? Then I feed in the masked part of that representation into my model, and then I ask my model to then fill in the blanks, right? So in this case, I have this idea of like learning about AI is awesome. I've blacked out AI and awesome. Then I ask the model to fill in what next word comes here. And I get a loss function, normally some idea of cross entropy, where I say like, how good was that performance, right? How confident were you that that next word was actually awesome, right? And then I ask how good that prediction was, and I continue to train. Now in biology, people have done something very similar, right? So let's go to the single cell space. I'm not saying this is the right way to do it. This is how people are doing it. You can think about the same sort of situation where now I have genes, and I have the gene names, and I have their gene expression. I could put them in a sequence. For those who are really familiar with biology, you're probably like, well, what's the sequence order? There is no sequence order. Just put them in a sequence. You can think about different ways to do this. There are some people that rank them based on expression. So you go from the most highly expressed to the lowest highly expressed. There are some people that think about this with respect to binning. So I might bin things. So I say, I put them in bins, and I'll order the bins. Whatever the case might be, you still have gene names and expression, the same idea. You can tokenize them, and then you can mask certain parts out. And then I ask, again, in this given sequence, as I train my model, can you predict the next gene name and the next its expression, right? How good were you at doing that ability? And then we'll continue to rotate, right? Now, there are a lot of capabilities in certain modalities for which this regime is actually really good, right? And some of this for which this makes sense. And a lot of it has to do with like the type of data that you're looking at, right? So the two types that people typically think about this with are DNA and proteins. And mostly because it's like sequence, right? These are like, this mostly resembles text, right? And people are very familiar with protein language model stuff because of AlphaFold, right? And what's really interesting when thinking about this versus expression data is one, expression data is tabular. So that's a totally different type of data in and of itself, but also, you know, there's a ton of data that I can get for both of these regimes, for proteins and DNA, right? I have whole genome referencing maps. I have like disease cancer maps for proteins. I can use synthetic data because I really understand the physics for how protein sequences are generated. And then there's a lot of functions for which I totally understand, for which I would have a foundation model do, right? If I had a good DNA foundation model, I might try to predict enhancers along the genome. Right? For proteins, it's really about coming up with new drugs, thinking about how to create new enzymes and new antibodies, right? That's like the promise of AI in this space. For single cell, we don't really have any of this. So this idea of, you know, what is a really good data set, you know, there are atlases that are coming out, but again, on the order of like, we'll talk about this in a little bit, but on the order of magnitude that you can get for these other data sets, we're still not quite in our infancy. And the real question about what you would have a foundation model do is also kind of a question mark. Like, what is it if you had a foundation model, what would it do? And I think some of us are kind of settling on this idea of perturbation prediction, right? I've seen cells in a given state, and I want to predict what happens to that cell gene expression, given that I've applied, let's say, some sort of drug, right? Some kind of like in silico type of assay, right? So, you know, despite this question mark being here, it hasn't stopped the field from trying. Actually, there's probably been like 4 that came out today on like new foundation models that people said that they figured this out, right? And there are 2 things I want everyone to kind of get from this slide. The first is that there are many that are coming out every single day, and they all take very different strategies. So some of them are transformer-based, some of them are autoencoder type-based, right? Some of them leverage data. UCE leverages data from protein atlases. And so that's one thing to kind of keep in mind, is that each of these models claim they're foundation models and that they all do very different things from very different standpoints. And the kicker is, a lot of times they don't also give out their pre-training code, so it's almost impossible to actually benchmark across all of them. So that's another thing. The second thing that I think is also a really key thing is the size of the data sets that they're trained on, right? This data, Geneform is trained on 30 million cells, UCE 36, SCGPT 33 million, SC Foundation 50 million. Last week I saw a model that said we trained a model on 150 million cells, right? And that number keeps getting larger and larger. And I get it, the incentivization from like a Nature Science Cell publication is that you train your model on the biggest thing, you'll get the best paper, right? But there's a question of like, why are we training data on such large data corpuses? So we started asking some questions. And I'll start with this idea of, I can imagine a world where fine-tuning, some of these models will be really great in the fine-tuning case, right? I train a model, I have a little bit of data, I update those model weights towards what I want it to do, and then I have it do that thing. So I can get where fine-tuning would be a good study. But we started asking a question like, what are the actual zero-shot capabilities of these models, right? And we broke this down into 3 basic questions. So this is a study done from an intern at Microsoft Research with us a couple years ago, Kasha, who's now a scientist at the Allen Institute, where she thought about a few things, right? In these types of, pre-trained type foundation models, do these models learn like data distributions and representations really well? Do their embeddings and capture actual biological variation, or do their embeddings just capture kind of like technical variation? And here's maybe where I start to see him as like a skeptic of these methods and not, but hopefully I'll end on a happy note. Okay, so here's an example of what I mean by this issue. So what SCGPT does, this is the model that we're going to evaluate here. SCGPT takes every single expression and it bins them, right? So from highest to lowest, it puts them in these bins. And you feed in those bins, and what you're supposed to be able to recapitulate is the order for which those cells or those genes appear in these given bins, right? And so here's your input bins, the distribution, all the way up to like 50, and here are the outputs, and really what you should see is this nice diagonal line, right, if I'm learning what I should be learning in my given model. But here what you actually see is that SCGPT has a very hard time actually learning like the lowly expressed bins, or actually de-differentiating between them. And it has some idea of learning like what is kind of highly expressed, but like no real separation, right? And so that's concerning. So what we actually did was think about, okay, well, if its pre-training task isn't going well, how well does it actually do in terms of prediction? Okay, so here are some results of prediction-based tasks. The prediction is actually being able to predict either it's going to predict gene expression, and we're going to do so from using the mass gene expression as a model directly, or I'm going to use I'm going to predict gene expression from like the cell embeddings that come from these methods. Okay. At the top, what you're seeing on the y-axis is mean squared error, so obviously lower means better. And this is my baseline model. So my baseline model, what I do is or what Kasha did, is we take the gene expression training data set, and I just take the mean of every gene across all cells. And that's my model. There's no training. I just take the mean gene expression. And I use that as a predictor of other gene expressions, and this is what you get. Okay? Now I'm going to take SCGPT and I'm going to feed it just junk. So I'm going to feed it like random noise. There's no real genes, it's just fake numbers. And then we'll have it trained, and we'll have to do a prediction, and it does really bad. So that's good. The concerning part is the rest of these, because the rest of these are actually really trained models on real data sets. So here we train SCGPT on just kidneys, just blood, and then human cells, so just like cells across different tissues. And none of them actually do much better than predicting the mean, if I just took this mean model, right? I was like, okay, so that's something. And so let's ask a further question. Like, actually, are these embeddings actually capturing biological variation, or do they even remove like technical variation? I mean technical because each of these cells in my data set were coming from different types of experiments across different technologies. So there's batch effects everywhere, okay? So here's a result of this. Again, let me orient everybody. On the left-hand side, you're seeing genes across, these are the total data are given on a UMAP, where each thing is color-coded based on cell type, right? Here's highly variable genes. So basically to get a good clustering, all of the different cell types of a given type should be clustered together. Highly variable genes means something also very simple. We take the total cell X gene matrix, and what I do is I just look for the genes that have most variation. So like I just calculate how variable they are across all cells. We draw a line at like 1,000, and then that's my model, right? And then we use that to do clustering with, and that's what my highly variable genes get. SCVI is also quite good at this, and SCVI is a basic autoencoder, so nothing, no bells and whistles, none of that stuff. And here are your two quote-unquote foundation models, GeneFormer and SCGPT. SCGPT has a hard time kind of modeling and clustering certain cell types together, and GeneFormer is kind of like all over the place. And so you start to ask, like, okay, well, what is it actually clustering? And the answer to that for at least GeneFormer and SCGPT is it's highly batch effects. And so here are the same experiments, now color-coded based on the technology that was used in the sequencer. So if I was doing a good integration task, you should see all the colors kind of intermixed together, right? They shouldn't all be separable. The highly variable gene model, which is no training at all, definitely does a really good job of this. We kind of just see this intermixing. SCVI also does a really good job of this. You see this intermixing. GeneFormer, which was all over the place up here in terms of biological variation, actually tends just to be learning just the technical variation. And SCGPT also tends to have a little bit of trouble and also learning this technical variation. So going back to the things that Kasha was asking at the very beginning, you know, are these models capturing the data distribution well? No. Do the embeddings capture this desired biological variation? Like, sort of, and they're basically just learning a lot of this undesired technical variation. So then I start asking a larger question. Is data actually a bottleneck? Because remember, in proteins and DNA, I have a lot of data, and you see a lot more promise maybe with these methods. So is data kind of the bottleneck here? And remember, a lot of these data sets and these models are being trained on like 30 million, 50 million, 150 million cells. And I have this question of like, why are we doing this? So we tested this. So this is driven by Alan in a paper that they were currently doing revisions on now, where we asked this, if we took 22 million cells, so part of the corpus from cell X gene, and I really asked, do I need all 22 million cells in order to see maximum performance? Like, what's the issue with doing these large data sets? So we did this experiment where we took 22 million cells and we downsampled them into data sets of size 1% of the data, 10% of the size, 25, 50, and 75% of the size. And we do this in 3 different ways. One is just kind of uniform random downsample. So I just downsample so I get 1% of the data and so on. I do another case where I downsample till I have 1% of the data, but I do so that I maintain the distribution of the cell types. So I know which cell types are there, and I want the same distribution of the cell types in my 1% data. I want that same representation. And then we do another case with geometric sketching, which is very similar to cell types, but instead of using the hand annotated labels, I just do a clustering algorithm over them, and then we do that downsampling. Okay? Now, what you're going to see on the right, on your right-hand side is a result where on the y-axis you're going to see performance. On the x-axis you're going to see the cell type, this distribution increase from 1% all the way to 100%. And what you'd expect to see is that as that number goes up, I'm going to see this increasing performance, right, in terms of how well I'm doing. And that's not what you see. And so what's actually really interesting is that, so again, here's your different downsampling schemes. So 2 things are really interesting here. The first is that it doesn't really matter how you downsample. It kind of all has the same type of variation that's involved. The second thing that's also really interesting is that everything kind of saturates at 1% of the data. And you might ask yourself, well, does this actually replicate for all these type of models? And it actually does. And so we did this idea where we said, okay, let's take these different pre-trained type models across different types of methods, across these evaluations, and then let's ask, at what point do I see this kind of saturation hit, right? At what point does the model look like it's actually not learning anymore as my data sets continue to grow? So we're gonna call that like our saturation learning point. And actually tends for zero-shot classification evals, you only need about 25% of that 22 million, right, to actually see that model not be able to learn anymore. So that's our zero shot. It's way worse for fine-tuning. For fine-tuning, if you're going to fine-tune, just take 1% of the data and then move on with your life, right? So then I really started asking another question. Okay, so the secret sauce in like GPT, right? A lot of people think it's the architecture, but the architecture is everywhere. So how come certain models can be better than other models? And a lot of it has to do with like the gas you put in the car, right? It's the training data set. It's the data set and how you introduce certain concepts to the model, where you want data sets in language. People have done a lot of studies on this, where I want data sources that are both similar but also orthogonal in terms of like the information they represent. That allows me then to cover enough space to generalize out of distribution, right? So these data corpuses are really, really important. And what we're doing in single cell right now is I'm just data dumping, right? I have 150 million cells, I put them all into the data set, and I continue to put as much as I want, but I'm not really learning about what cell types are enabling me to learn about other different cell types, right? And so we did an experiment of like, how valuable is this to leverage biological diversity? And one way you could do this, someone talked about the hematopoietic stem cell system yesterday, so I really appreciate that because I don't have to go over too much of it in detail. But one thing you do is you can leverage the idea that like cell types are related to each other, right, in a given tree, right? And if I really had a data set that was all-encompassing, the way I would do it is I would create a training corpus that kind of, in concert together, kind of tessellate all the way across this tree where I have like all these different cell types, right, kind of covered across all these cell states, right? And so let me just give you an example of like why this is really important. So let's take an example. This is a thing done by my intern last year, Ajay, where if I take a model and I train it on just blood cells, right? So I'm going to just cover maybe this portion of the tree. I'm going to train a model just on 230 healthy blood cells. And then let's take another model and let's train it on 115 healthy blood cells. But then let's also take a model on that transcription factor data set I was talking about at the very beginning of my talk, where I did these ORF screens. And let's train it on 115,000 cells come from that type of experiment, right? And the idea is that they're going to cover totally different cell states, right? One's a perturbation and one's just like healthy state. Now, if my test set is in blood, so let's say I want to predict the expression of new blood cells. Well, I should do just fine, right? Because I've seen blood cells. Both of my models have blood cells in them in the training set, so I should be able to generalize really well to blood. And that's what you see here. On the y-axis, you see R squared, so higher means better. And both my models, whether I'm trained on just blood or blood plus the TF atlas, I'm doing kind of the same. Now, let's say I want to test my model and predict the expression of neurons. So I've never seen neurons before in either data set, right? And so you get something that looks like this, where my blood model has no way that it can ever generalize to neurons because it's too far outside of the states that data set covered. It's too narrow. What you're going to see is the R squared very low, right? But at least with the transcription factor cells, they generalize at least out of distribution more than just the blood that allow me to get some sort of explainability back, right? And this idea is actually really important because you start to ask yourself, does this generalize out of distribution? Like, how valuable is it to think about these different types of perturbations, right? And actually the key is like, it's actually really important. So whether I'm looking at trying to predict neurons, kidney cells, or heart cells, having that TF atlas involved always is a huge help, right? And the reason for that is because that the TF atlas just expresses more different states than any of these other types of data sets, right? So these perturb-seq experiments, these experiments where you can go in and you can perturb the cell in some sort of way, or these TF atlases where you're kind of engineering them, not just with drug, but with something else, with these like ORF type screens, you can get more states that are represented, which is really nice because they start to cover things that are just not explainable by other models. So what I can do is I can look at the embedding from my blood model and ask which types of embeddings from the neurons does the blood model struggle to explain? And it's something like this, but that's highly explained in my TF atlas, right? And so I can start to explain sources of variation that I just couldn't do before. And so the idea, again, in Ex Vivo, to bring it all back for those last couple of minutes, is we can actually engineer this heterogeneity, right? And so we're trying to actually do this. So, I have a transcription factor library, like this stuff that's being led by Evelyn, where what we're doing in Ex Vivo is I'm trying to build these data sets where I have enough diversities that my models can learn from, so I really start to generalize out of distribution. And the key part here is it's not about the number of cells I have. It's about the diversity of the cell states that are covered. So my data sets can be small, but as long as they're information rich, that's all I actually really care about. And so what we're trying to do now is across many different types of engineered heterogeneity, across many different hallmark perturbations, across different tissues, right, start to cover this entire state group. Now, if you're looking at this like my Microsoft leadership did when I first pitched this idea to them, you might just be like, that's a lot of dollar signs. Experiments cost a lot of money. Like, who's paying for this? But the really beautiful part about Ajay's project is it kind of teaches us how to move across this in a more information theoretic way because of this given connection. So here's a larger picture from Ajay's project, and it seems like a lot, but I'll break it down really quickly. What we do here is we train different models across different sources of training data. So we take a blood data set, a bone marrow data set, a cancer one, a perturb-seq, and a transcription factor atlas, and then we do combinations with them where we combine blood with one of these four. And then we test them to be able to predict gene expression in another blood data set, something in distribution, a bone marrow, 2 different cancer types, and then neurons. And what you're seeing here is blue means that that model did worse than just the baseline, and green means that you see some improvement. And what I actually learned from this is that you don't actually have to see disease states in order to be able to generalize to disease states. And what's really interesting about this is if I did my model in blood and I wanted to then predict something like AML, adding cancer data doesn't actually give me a massive, huge bump in terms of performance, right? What you actually really care about are these settings where, again, for the TF atlas, something's very far out of distribution. Why that's important for us experimentally is because what that means, if I go back to this thing that would freak out Microsoft leadership, what I could say now is that, well, let's say I would do a perturbation on one part of this, where I do like a KRAS inhibitor on one tissue type. That might actually tell me what it means to do, let's say, interferon gamma perturbation in another tissue type. So this idea that certain states will generalize and give me information for free about other ones, right? And that's actually you really see in real life, right? There's this idea of certain cells that'll have certain like cell type specific fitnesses, but there are a lot of them might have shared fitness effects. And that's really important because that now allows me how to sample around this a lot smartly, right? And so now we could build AI models to really learn links between both cell states and their function. And so this is also what we're doing at Ex Vivo, this idea of building new data sets and building new models. But in this flywheel, the really important key parts here are this idea of like when I have new data, triaging to be like I have a lot of new cells, but they don't actually give me any new information. And so this triage step is actually quite important where we're doing a lot of work. In the last 2 minutes, what I'll say is, you know, one thing that we've been doing a lot is one thing someone might ask is like, is cell state just represented by RNA? And the answer to that is probably no, right? And what everybody's moving towards is this idea of multimodal learning, right? Stuff that you'll see in a lot of different areas where you have like vision and text, right? A lot of stuff that you'll see in these like new vision models. Same thing is happening in biology. We're thinking about how to go across different modalities. So one thing that we've been doing is thinking about this idea of, again, information richness. When I have different data sets come from 2 different modalities, let's say imaging and gene expression, at what point is that information contained in both those data sets unique, redundant, or even synergistic? And the synergistic part is the one I really want to hit at, right? It's the part where, like, without those 2 modalities together, I would never be able to generalize for this new task, right? So I have an intern this year actually thinking about this question, Till Richer, where we've been taking data sets from H&E slides where we have both image for them as well as spatial transcriptomics, and thinking about, you know, if I ran these through these individual, like, you know, foundation models, and then thought about how to align them in some way, at what point do I get this kind of synergistic score kind of information where I have information that is coming from just, that without having both those modalities together, I wouldn't have seen this like increased predictive ability, right? And so we've been doing this across different tasks where they might be kind of classified different spots and niches to doing cell type composition. So within each of these given spots, trying to understand what cell types are present, or then trying to predict for like neighboring slides effectively. So with that, I'll say, yeah, we have these 2 different aims. As a group, we are always interested to chat with people and do further collaborations if anyone's ever interested. It's been a really fun thing. There are acknowledgments, people have given us support, and always thank you to the patient and families that have been part of this study. Yeah, I'll have to take any questions at all. Appreciate it.
Going back
Hi, thank you for the talk. So you mentioned like the data sets are like 30 million or like 15 million, but then I would imagine that's like, even though like it's in the millions, it's pretty small in scale compared to like ChatGPT or something, because like they train it on like the entire internet. And then I mean, I don't know about people, but like to me, like predicting cells and like genomics is like much harder than like predicting the next word and like the token. So could it be like a problem that like you don't have enough data to like, feed on it to like do like another version of like the scaling hypothesis for sales? Or like, is it like an algorithmic problem, or like what's the bottleneck here? Like,
yeah, I think sorry. So I think there are a few issues here. First is, like you mentioned, okay, so right, the data sets can be, so you have data sets that are like think on the order of like, so you have 100 million cells, you have 20,000 genes. The issue is that single cell data is quite sparse, right? So a lot of those numbers end up being zero because many of those genes are like not expressed. The second issue is they're super low rank. So out of those 20,000 genes, I only need about like a couple hundred, if that, to really explain certain like large axes of variation across cell types, right? So then it becomes a question of a few things. One is architecturally, is a transformer the right architecture? Because we're in like this tabular thing, and I don't know, I think people are investigating this more heavily now, but before it was like, well, this worked for proteins, and it got someone a Nobel, right? Let's also do this for cells, and I don't know if that's the right conversion. So people are also asking for this tabular type data, should we be doing that? So that's one question. The second question is again, I think goes back to training objectives, right? And we're also trying to fit this kind of mass language modeling objective, but the idea of like genes don't have a natural like sequence order. So is predicting the next gene in a random sequence also the right way to learn something? Or should it be a different training objective? And then to me, the third question, which no one really thought about, is are we actually asking these models to do a task that would require the sophistication that these larger transformer models would be used for, right? So in other words, like the cell type annotation task, I did quite fine with like a linear model, right? But so why do I need to throw a large transformer for it? So I guess it's a question of like scaling task complexity. Is there a point at which the task becomes so complex that the sophistication of these architectures then kick in, where I don't actually need these simple models anymore, and these larger models are much more necessary. So I think it's a multifaceted question. I don't have one single answer for you, but I think it's like all those factors.
Okay. Hello. Thanks for the informative talk. I would also like to ask the question, second the first question, and given your answer. So given the sparse data, I also believe like for rare cancers and when we have tens and thousands of genes, do you also consider like dimensionality reduction? Because and that was one point I wanted to raise. And the other one also in the dataset paper part of your research, like while you were doing data cleaning, are you also considering normalizing the corpus?
Yeah, great question. So first, the second one's easy. They're always normalized. So yeah, all the data sets are always normalized. Then the first question was about dimensional reduction. So, okay, so what people tend to do is because they assume that most of the 20,000 genes are not necessary, what a lot of people do is they'll work off highly variable genes. So what they'll do is they'll reduce the number of genes that they even look at, even at the very onset, to like, 1,000 to 5,000 genes, right? There are a few maybe issues with that. One is like the data set that you do your dimensionality reduction on from the highly variable genes perspective might not be the same highly variable genes that matter for the data set you're trying to generalize to, right? So what if I try to generalize to a new cell type, but the cell types that I did my dimensionality reduction on, they just have two different signatures. So that could be an issue. So the question is like, what goes into that 5,000 set, right? And in terms of like how people do the data management, so how they build those embeddings, that goes back to the architectural question, I think, which is like, what's the best way to think about how to generalize for those embeddings? And I think that's like a modeler's choice thing that's still like ongoing research for a lot of people.
Okay, thank you.
Thank you for the talk. It's very interesting that the diversity kind of really plays an important role in clustering. I'm just wondering, is there a way to measure the diversity of a data set that may be predictive of out of distribution performance? Is it the information content or the variation? Can you talk more about this?
No, this is great because I have an intern doing this right now. Actually, we should talk. hopefully she won't be mad at me telling her project to everybody. No, this is exactly what you want to do. So there are a few ways to think about this. One is from a task perspective, which is like, can I think about synergy across data sets conditioned on task? That's less interesting because what you really want to do, I think, that measurement of data diversity is from like intrinsic perspective of like the data sets are just what they are. So there are a few ways to think about this, and I'll just tell you the ones that like we're trying to explore now. One is thinking about like, distance. So in an active learning setting, I have a data set that I've collected, and I have a new data set that's come online, and I basically want to know, like, is that data set worth adding to my corpus or not? There are a few ways you could think about doing this. One is like from like almost like an optimal transport sort of way, where you can ask like, how much energy might it take for me to move from the data set I have in my training set to the one that I just collected off the sequencer? And if that is high, then I'm like, oh, I can't do that, so that's information rich, I'll keep it. There are the more information theoretic ways, which are thinking about like mutual information and these sorts of things, which is probably supernatural. There's a ton. I don't know if people are thinking about it from this way though. Where I think people in this room might really think about it that way. I think everyone else in the field, what they're doing is saying, that adds to my millions, and so I'll continue to add with this idea that like scaling laws should kick in, but I don't think the typical scaling laws that you will see in other regimes kind of factor here. I think the scale is a little bit different. Yeah. Anyway, I'm happy to, I'm actively thinking about this, so yeah.